Hello, my name is Renee Rachi, and I'm the Commercial Product Manager for Custom Oligos and qPCR at IDT. I will be your moderator for today's webinar. I'm really excited to have Dr. Sarah Beaudoin present to you one-step qPCR master mix for amplification from crude or purified samples. This presentation gives you features and benefits of our new innovative qPCR master mix called PrimeTime One-Step 4X Broad Range Master Mix. Dr. Beaudoin is a staff scientist in the Enzyme Evolution Research Group at IDT. Sarah graduated from Georgia Southern University in 2012 with a BS in chemistry, where she was an undergraduate research fellow working on biocatalytic synthesis of chirolactones. She earned her PhD in biochemistry from the University of Florida in 2017, where her research focus was the synthesis of non-natural amino acids using threonine analytes. She joined IDT in 2018, and her recent work has focused on reverse transcriptase evolution and master mix development. We welcome your questions, so please type them into the chat, and we will address as many as possible after the presentation as time permits. And now I will hand it off to Dr. Beaudoin.
Thanks, Renee. In this webinar, we will discuss the basics of RT-qPCR, specifically as it pertains to viral identification given the recent pandemic crisis. While IDT has been producing primer and probe sets for the identification of SARS-CoV-2 and influenza that include sequences recommended in the CDC protocol, we had not previously offered a master mix that allowed for one-step identification of viral genomic material. Today, I'll introduce the solution to that problem, the PrimeTime One-Step 4X Broad Range Master Mix. I'll describe the development process and discuss characteristics and general usage information. And finally, I'll show data using multiplexed primers and probes specific for influenza A, influenza B, SARS-CoV-2, and RNase-P for direct amplification of viral RNA in human specimens. IDT has always been known for high-quality primers and probes used in qPCR workflows. For example, we were one of the first to market for primer probe sets used as a component for the identification of COVID-19. IDT has not been as well known as a provider of master mixes. In September, IDT released another component that can be used in viral identification. The PrimeTime One-Step 4X Broad Range Master Mix directly converts the viral RNA genome into DNA for identification by qPCR. This slide depicts an example of a current qPCR workflow used in COVID research. The nasopharyngeal swab is used to collect viral particles. The viral RNA is isolated from the sample using an extraction kit. The viral RNA is then converted to cDNA using reverse transcriptase that is included in the qPCR master mix. The qPCR reaction then makes copies of the cDNA and simultaneously identifies the presence of the copies via the hydrolysis of the fluorescent probes. Of note, IDT provides the primers and probes for this process, and we now offer a one-step master mix as another component of the identification of viral RNA. You can use this master mix for standard workflow after RNA extraction or direct amplification straight from VTN with the addition of the provided enhancer solution. The extraction components and thermocycler used in this process are provided by other vendors. IDT is offering two new one-step RT-qPCR master mixes. PrimeTime One-Step RT-qPCR Master Mix is a master mix we developed early on that is an excellent 2X formulated product with CTs comparable to other tested competitor master mixes for purified RNA templates. However, there are some desirable attributes that we were missing with this one-step RT-qPCR master mix, such as inhibitor resistance and formulation concentration. We acquired customer insight to gain a list of important customer needs. Today, we are introducing our new PrimeTime One-Step 4X Broad Range Master Mix. The primary goal attributes attained for this product are as follows. 1. 4X formulation. The new master mix is a 4X concentrate to allow for flexibility in sample loading. 2. Inhibitor resistance. The new master mix is inhibitor resistant and can handle high concentrations of known qPCR inhibitors such as heparin, hematin, and humic acid. 3. Low threshold cycle. The new master mix has equal or better CT to that of all other leading competitor one-step master mixes. 4. High endpoint fluorescence. This is something that was strongly communicated to us by many customers. The new PrimeTime one-step 4x broad range master mix was designed with this in mind, and in turn is a master mix that shows high endpoint fluorescence. Other tested competitors' one-step master mixes don't come close to the endpoint fluorescence achieved by our new master mix. A secondary and almost unattainable goal that we were tasked with was to provide a master mix capable of direct amplification of specimens out of VTM. Our new PrimeTime one-step 4x broad range master mix with direct amplification enhancer is compatible with RNA templates contaminated with highly inhibitory human specimens such as those from nasopharyngeal and saliva swabs in VTM. I will be going into more detail on all these attributes throughout today's webinar. The PrimeTime one-step 4x broad range master mix's improvements come from a combination of an improved buffer and proprietary mutant enzymes. The R&D efforts for the buffer formulation were exhaustive as we iteratively screened approximately 35,000 different conditions and combinations through exceedingly complex high-throughput screens to achieve our highly innovative and inhibitor-resistant master mix while maintaining the capability of a 4x formulation. We wanted to focus on improvements to the DNA polymerase and the reverse transcriptase. So R&D efforts continued through enzyme high-throughput screening to obtain proprietary mutants of both the reverse transcriptase and Taq polymerase. That allowed for specific characteristics such as increased activity, inhibitor resistance, threshold cycle, and endpoint fluorescence. Further high-throughput screens were performed to develop a companion product called the direct amplification enhancer that, when added in combination with the PrimeTime one-step 4x broad range master mix, allows for a boost in amplification of RNA templates contaminated with highly inhibitory human specimens such as nasopharyngeal and saliva matrices in VTM. The kit also includes a separate tube of ROX dye standards for systems that require this. As mentioned, our new PrimeTime one-step 4x broad range master mix has high endpoint fluorescence. These data depict the results from RT-qPCR assays containing multiplexed primers and probes specific for influenza A, influenza B, SARS-CoV-2, and RNase P genes were used to amplify pooled RNA ultramer templates ranging from 1 million copies shown in red down to 10 copies shown in blue. The data for influenza B is shown. Both the linear and log amplification plots are shown for our PrimeTime one-step 4x broad range master mix displayed on the top panel, and a leading competitor's master mix shown on the bottom. Our PrimeTime one-step 4x broad range master mix has significantly improved endpoint fluorescence compared to the competitor master mix. This is particularly important for lower copies of RNA template, where you typically see a drop in endpoint fluorescence, and sometimes signal can be lost in the noise. IDT's PrimeTime one-step 4x broad range master mix provides exceptional multiplexed RNA identification up to a 5-plex over 8 logs of purified RNA template. These data depict the results from a multiplexed assay where we used pooled RNA ultramer templates ranging from 10 to the 8 copies down to 10 copies that corresponds to 5 genes commonly expressed in human cells: CHEK1, CSK, HPRT, RNase P, and SFRS9. The CHEK1 primers and probe set is being used on the left. The left panel shows the delta RN plot of a dilution series of CHEK1 RNA. The panel shows the standard curve plot demonstrating that the master mix performs well with a correlation coefficient of 1 and an efficiency of 97%. In the middle and right panels, the CSK, HPRT, RNase P, and SFRS9 are the primer probe sets, and TAMRA, FAM, Sci-Fi, and SUN are the identification dyes in the multiplexed reaction. Like on the left, the amplification plots and standard curve graph show the master mix is performing as expected. IDT's PrimeTime one-step 4x broad range master mix provides excellent multiplexed limit of detection when using purified RNA as template. These data depict the results from RT-qPCR assays containing singleplex or multiplex primers and probes specific for influenza A, influenza B, SARS-CoV-2, and RNase P genes were used to amplify 100, 50, 10, 5, or 1 copies per reaction of purified RNA template. A full analysis of results is displayed on the left with a representative amplification plot for the multiplex SARS-CoV-2 displayed on the right. RT-qPCR reactions containing 5 copies of each RNA template with either singleplex or multiplexed primers and probes amplified 100% of the reactions, where 21 reactions were monitored. The new PrimeTime one-step 4x broad-range master mix provides excellent resistance to common qPCR inhibitors as compared to 3 other commercial master mixes. These data depict the results from a multiplexed assay where we used purified RNA template in the presence of the indicated concentration of inhibitor. The left panel shows the change in threshold cycle between the RNA sample absent of heparin and the RNA sample contaminated with increasing amounts of heparin, where the smaller the value, the more resistant the master mix is to the indicated inhibitor. Each master mix was used precisely according to each manufacturer's instructions. At any given concentration of heparin, the new PrimeTime one-step 4x broad-range master mix is minimally affected. We also see a similar trend for hematin, data shown in the center panel. Humic acid, shown on the right, shows the new PrimeTime one-step 4x broad-range master mix is less affected as compared to 2 of the 3 inhibitors. One of the most sought-after requests from customers is to offer an extractionless direct amplification capability in a one-step RT-qPCR master mix. Currently, RNA extraction is recommended for identification of analytes contaminated with human specimens before RT-qPCR due to many challenges. First, the viral capsid must be denatured either through chemical denaturants or heat treatment to release the RNA from the protective coating. Both viral and heat denaturation methods kill reverse transcriptase activity. Second, human specimen samples are stored in viral transport medium. This composition is highly variable and is generally inhibitory to qPCR. And lastly, nasopharyngeal and saliva samples are highly inhibitory to qPCR as they are loaded with nucleases, proteases, and numerous inhibitory compounds. We sought to address these challenges when developing our IDT PrimeTime one-step 4x broad-range master mix. For viral capsid denaturation, we created a 2-step solution. The first was to combine the heat extraction and the reverse transcription into a single 15-minute initial 50 degrees Celsius step, and then proceed to qPCR. This was partially solved by our buffer composition, but also one of our proprietary mutant reverse transcriptases developed with improved activity in this context. These data depict the amplification curves with a gradient heat extraction step ranging from 50 to 74 degrees Celsius. The mutant reverse transcriptase on the right retains most activity when heated with various denaturation temperatures, whereas the wild-type reverse transcriptase on the left loses activity as temperature increases. Another challenge is human specimen samples are stored in viral transport medium. This composition is highly variable and is generally inhibitory to qPCR as it contains chemicals that denature proteins, among other things. We have found that these solutions are generally inhibitory to all qPCR master mixes that we have tested in-house. However, our new PrimeTime one-step 4x broad-range master mix is particularly resistant to multiple formulations of VTM. In all, we have tested more than 10 different formulations of VTM, and our new master mix stands up to them all. Another challenge is nasopharyngeal and saliva samples are highly inhibitory to qPCR as they are loaded with nucleases, proteases, and numerous inhibitory compounds. These sample types habitually reduce the lower input identification and endpoint fluorescence of RT-qPCR master mixes, and there is no cost-effective RNase inhibitor solutions on the market that are thermostable. These data depict the results from a multiplex assay where we used 30 copies of AccuPlex SARS-CoV-2, Flu-A, Flu-B, and RSV verification panel material as the template. On the left, the AccuPlex analyte verification panel is diluted in tris EDTA buffer to show baseline amplification as compared to in BTM, which is shown in green, in a human nasopharyngeal matrix in yellow, and lastly, a human saliva matrix on the right panel. As you can see, signal is substantially reduced in the presence of nasopharyngeal matrix and non-existent in the saliva matrix. These data display the challenge we see for these highly inhibitory sample types from human specimens. Our solution was to develop a direct amplification enhancer sold as a companion to our new PrimeTime one-step 4x broad-range master mix that, when added to the RT-qPCR reactions, negates most of the inhibitory effects observed when human specimen matrices are the template. These data depict the results from a multiplex assay where we used 30 copies of the AccuPlex analyte verification panel that was diluted in human nasopharyngeal matrix as the template. Amplification of material by the new IDT PrimeTime one-step 4x broad-range master mix without direct amplification enhancer is displayed in red and shows the expected result for all 4 targets, which is a drop in signal and endpoint fluorescence in the presence of human NP matrix. However, our new master mix with our direct amplification enhancer, which is shown in yellow, recovers both signal and endpoint fluorescence in the presence of the inhibitory human NP matrix. This same result is also observed for samples containing human saliva matrix, with an even more dramatic improvement in signal and endpoint fluorescence with our new master mix and direct amplification enhancer shown in yellow. IDT PrimeTime one-step 4x broad-range master mix demonstrates comparable amplification of viral analytes between extracted and non-extracted human samples. These data depict the results from a multiplex assay where we used 30 copies of AccuPlex SARS-CoV-2, Flu-A, Flu-B, and RSV verification panel material that had been either extracted in blue or non-extracted in gray as the template. For the extracted RNA samples shown in blue, the AccuPlex analyte verification panel was first diluted into negative human saliva or NP matrix, followed by automated extraction using the Kingfisher Flex 96 and MagMax Viral Pathogen 2 nucleic acid isolation kit. The resulting RNA was added to the new PrimeTime one-step 4x broad-range master mix as the RNA template, and results indicated as the blue amplification curve for saliva and NP samples. For the non-extracted RNA samples shown in gray, the AccuPlex analyte verification panel was diluted into negative human saliva or nasopharyngeal matrix and directly added to the new master mix with our direct amplification enhancer. As shown, RNA samples resulting from non-extracted human samples using the direct amplification method, shown in gray, actually have increased endpoint fluorescence compared to extracted RNA, which is shown in blue. This is likely from the carryover of ethanol, guanidinium thiocyanic acid, and/or beads commonly seen from these types of extraction kits. Both extracted and non-extracted RNA were amplified using the QuantStudio 7 Flex instrument, and amplification curves for RNA from human saliva are shown on the left panel, and human nasopharyngeal shown on the right. A similar assessment of the limit of detection was performed for direct amplification in NP matrix. RT-qPCR assays containing singleplex or multiplexed primers and probes specific for influenza A, influenza B, SARS-CoV-2, and RNase-P genes were used to amplify 20, 10, 5, or 1 copies per reaction of AccuPlex SARS-CoV-2, Flu-A, Flu-B, and RSV verification panel material diluted into negative human nasopharyngeal matrix. A full analysis of the results is displayed on the bottom with representative amplification plots, multiplexed SARS-CoV-2 primers and probes displayed on the top. RT-qPCR reactions containing 10 copies of AccuPlex analyte verification panel in the presence of human nasopharyngeal matrix is detectable. We also investigated this limit of detection for AccuPlex analyte verification panel in the presence of human saliva matrix, and find that the RT-qPCR reactions containing 10 copies of AccuPlex material is also detectable. And finally, IDT's PrimeTime one-step 4x broad-range master mix provides superior analyte identification from human saliva and nasopharyngeal matrix as compared to competitors. These data depict the results from RT-qPCR assays containing multiplexed primers and probes specific for influenza A, influenza B, SARS-CoV-2, and RNA-sp genes were used to amplify 30 copies of AccuPlex analyte verification panel spiked into negative human nasopharyngeal, shown on the left, or saliva matrix, shown on the right, stored in viral transport medium. IDT PrimeTime one-step 4x broad-range master mix with direct amplification enhancer, indicated in dark blue, is far superior for amplification, both threshold cycle and endpoint fluorescence, of non-extracted RNA samples in both human nasopharyngeal and saliva matrix compared to competitor master mixes. In conclusion, IDT PrimeTime one-step 4x broad-range master mix demonstrates exceptional capabilities as a multiplexed RNA identification master mix for both purified RNA templates and direct amplification of analytes in human specimen. It has excellent dynamic range for synthetic RNA templates of 1 to the 8 down to 10 copies per reaction. It has massive multiplexability of up to 5 RNA targets in a single reaction. It has high endpoint fluorescence and exceptional CT values for increased identification of low-copy analytes. It has excellent inhibitor resistance to common qPCR inhibitors such as heparin, hematin, and humic acid. And lastly, it has unique direct amplification capabilities that allow for identification of RNA targets straight from nasopharyngeal or saliva swabs in VTM, rather than being extracted prior to RT-qPCR. Our new PrimeTime one-step 4x broad-range master mix is currently available in various sample sizes to meet your research needs.
Thank you so much, Sarah, for that informative presentation. We're really excited about this new product and its flexibility to give researchers choices to optimize their qPCR workflows. At this time, we will answer some of the questions that came through the chat. Sarah, the first question is, can the direct amplification enhancer be used with purified RNA samples?
We do not recommend the direct amplification enhancer to be used for purified RNA samples. And that's because we see a small drop in the sensitivity at very low copy numbers for more difficult primer and probe assays.
Okay, the second question is the master mix compatible with different real-time PCR instruments?
Yes, the data shown today were generated using the QuantStudio 7 Flex, and the master mix has also been successfully used on the Biorad CFX 384, the Roche Light Cycler 480, and the QuantStudio DX in the RUO mode.
Great. Can the master mix be used in direct amplification from other human specimens, such as blood?
Although we have shown this master mix has high tolerance towards hematin and heparin, we have not tested amplification from blood, either human or other species.
And the last question I have that we have time for today is, how long can I leave the master mix at room temperature?
So without the addition of template, the qPCR mix can be stored at room temperature for less than 2 hours or at 4C for up to 16 hours.
Awesome. Thank you so much, Sarah. That's all the time we have for today. Thank you so much for attending this webinar. If you have questions that we did not address, please contact us at www.idtdna.com/contact us.